So last time we left off talking about Lazzaro Spallanzani. Remember, he repeated John Needham's experiment where he boiled his broth in the flask. He boiled it long enough, he sealed the flask, and nothing grew. So Spallanzani did the experiment correctly, and his results demonstrated biogenesis because nothing grew, so there's no spontaneous generation. But John Needham was much more popular, much more powerful. Lots of people believed him. So nobody really believes Spallanzani. So most people still consider that microorganisms magically appeared, due to some, magical vital force, even though Spallanzani did everything correctly. And it took over 100 years, or about 100 years, for Louis Pasteur to finally conclusively demonstrate that microorganisms come from pre-existing microorganisms. And he did the same kind of experiment that Spallanzani and Needham did, but he had a special flask. So let's look at that flask. So his flask, he filled his broth. It had a long glass neck, which he bent by heating it up, making an S-shaped or a swan-necked flask. Then he boiled his broth at least 30 minutes, making sure he killed everything in it. And then he let his flask sit, and day after day, week after week, even month after month, the broth was crystal clear. Now, Pasteur's flask was actually open to the environment, so oxygen could get in, right? The magical vital force could get in to cause spontaneous generation, but nothing ever grew in his flask until Pasteur did something. When he took his flask and he tilted it sideways and allowed the broth to run down into the neck and then go back into the flask, in a few days the broth was all cloudy and full of microorganisms. Where did they come from? So when Pasteur turned the flask sideways, let the broth run into the neck and then back into the flask, microorganisms started growing. So how did they get there? Where did they come from?
The air. From the air.
From the air, or as Christina said, from the dust and particles, the spores that were landed in the neck of the flask. Since it was open, to the air, and there's stuff in the air, we already learned that. That stuff just kind of landed in the neck, but it couldn't get into the flask until Pasteur physically turned the flask and contaminated broth. So the microbes were in the neck, and when he contaminated the broth with them, then they grew. So Pasteur finally conclusively demonstrated that microorganisms come from pre-existing microorganisms, so biogenesis. Now, Pasteur did not simply do this experiment once. He did it over and over and over again, not only to convince himself, but to convince everyone else, because people still believed in spontaneous generation. Finally, he was able to prove to everyone microorganisms already exist and they produce new microorganisms, therefore biogenesis. But look how long it took in order for humans to finally believe that microorganisms come from pre-existing microorganisms. It took a very long time. So began the golden age of microbiology, because around this period of time, we did have microscopes, so they could see bacteria. They had learned ways to cultivate, right, to grow the bacteria in the lab. They were starting to experiment with them. They were learning the relationship between microorganisms and disease and coming up with ways to prevent the spread of disease. They were learning about aseptic technique, sterile technique, especially for surgery. They were beginning to learn about the immune system, although it took a long time for them to put all the different parts together to come up with a cohesive explanation for the immune system. And they also began experimenting with different chemical compounds to see if they could use those chemicals to kill microorganisms in people. So that was the beginning of antimicrobial drugs. So a lot of incredible research was being done at this time by a lot of different people. So Pasteur is one of the most famous. He was French. Of course, the French do love their wine. And the problem with making wine is that sometimes it would work perfectly and they would get a nice alcoholic wine. And then other times the wine would be very acidic. It would turn to vinegar and therefore not be good wine. And of course, the winemakers did not know what the problem was, and they actually called on Pasteur to come and help them. And one of the things he told them is that if you have a bad batch of wine, clean all of the equipment before you reuse it, because that's not something that they, regularly did. But he said, make sure that everything is nice and clean before you reuse it. And Pasteur was the one who determined that a microorganism was actually producing the alcohol. Now, there was a German at the time, and Pasteur did not like him because he didn't like Germans in general, but that's another story. And the German, he said that alcohol was simply the result of the chemical breakdown of things like proteins. So Pasteur was able to demonstrate that no, it was a microorganism. Technically, it was a yeast, a eukaryotic single-cell microorganism, but Pasteur didn't know that at the time. He just knew it was a microorganism. And he showed that yeast produced the alcohol because when you grow the yeast with only sugar, no other protein or anything present, then the yeast produced the alcohol. So it's not a simple chemical breakdown. So Pasteur, he determined that microorganisms were capable of fermentation. He also determined that microorganisms were capable of spoiling food. And because microorganisms do spoil food, Pasteur came up with the technique of heating that food or that product just enough to kill many of the microorganisms, especially any potential pathogens, without destroying the product. And this process bears his name, pasteurization. So, for example, milk. Milk is pasteurized. It is not sterilized. There are bacteria in the milk, no pathogens, but there are bacteria present. Even if you never open the milk, it will still go bad if you give it enough time, because bacteria are already there. So how many of you worry about the expiration date? Or maybe your mother worries about the expiration date on milk.
Yes, I do.
Anybody throw their milk when it reaches the expiration date?
Yes. Yeah.
Why do you do that? That expiration date doesn't mean much. So the expiration date is just something they have to do for legal purposes, but milk can last well beyond the expiration date. And the way to determine that is not to open it and smell the carton. It's to actually pour milk in a glass and then smell it. Because bacteria start growing on the inside walls of the carton, so that's what you'll smell. And the milk may be perfectly fine. So always pour some into a glass and smell it. If it smells fine, then it is fine. And even if your milk starts to develop a little bit of what I call a bite to it, it's still safe to drink. Because spoiled milk is how you make yogurt and sour cream and buttermilk. So expiration date on milk doesn't really mean much. So pour your milk in a glass, smell it first. If it smells fine, it's good. So don't waste your milk. In the 1860s, Joseph Lister used a chemical disinfectant to help prevent surgical wound infections. So again, now people were starting to learn about the relationship between microorganisms and disease. And during surgery, as I mentioned before, it was not common for the surgeon to scrub, before surgery. And of course, they didn't have rubber gloves yet, so they didn't do that. And also really cleaning and disinfecting equipment, their surgical tools, that didn't happen either. But Lister discovered that if you can spray a fine mist of disinfectant over the surgical area, you could greatly reduce the number of surgical infections. And that was the beginning of using disinfectants in the surgical area. And you may recognize the name Lister. Look familiar? Listerine, right, Rachel? So Listerine originally was a disinfectant used to scrub tables and floors and stuff like that. It was not a mouthwash to begin with. So Robert Koch, or we would often say Koch, but he was German. In 1876, he was able to come up with a series of steps that could help determine the cause of a specific disease. So, and we'll talk about Koch's postulates later, but if you follow these series of steps, you should be able to conclusively demonstrate which organism is causing which disease. And we'll learn about that later, and also the problems associated with that. But going back to Lister in the 1860s, who can tell me what was going on in the 1860s in the United States? What was happening? In the 1860s. No, not disease, Christina. Those of you that have taken American history should know. No? Wounded people. Yeah, but how were they wounded? Right. Which war? The Civil War, correct. So the Civil War. So again, it was the 1860s. They were still learning about microorganisms and disease. So it was very common, when a soldier had a really bad wound and needed an arm or a leg amputated, they just put them on the table, take a saw, a dirty saw, and saw off the limb and bring in the next soldier and just use the same saw, right? They didn't quite understand that they should be cleaning and disinfecting. And as was the case in most wars, the soldiers or the warriors who died often died from infections they got after they were wounded, not from the actual wounds themselves. So now we had a way to reduce a number of those infections, right, by cleaning and disinfecting, washing hands. But that took a long time for people to accept that their hands are dirty, especially doctors. So a little bit before the time frame was Edward Jenner in 1796, and he gets the official credit for vaccination, even though human beings had, sort of been doing this kind of thing for a while. What he noticed is that in towns and villages that had a lot of smallpox, the milkmaids, the women who milked the cows, rarely got smallpox. What they did get was a mild infection from the cows called cowpox. And what Jenner correctly surmised is that the cowpox was protecting them from smallpox. And so he did what we would, sort of call clinical trials today, where he inoculated a bunch of people with cowpox, and then he observed, the people who were inoculated with cowpox and those that were not, how many of them got smallpox. And the ones that got inoculated with cowpox either did not get any smallpox or it was rare. Right? There were very few of them that did. So Jenner said that cowpox can protect against smallpox. And so began the process of vaccination. But Jenner was not the only one to notice this, but he's the one who documented it, so he gets the official credit. And prior to using cowpox, what people used to do to help protect against smallpox. Smallpox produces blisters, and when the blisters heal, there's a scab, and the scab actually contains virus, which theoretically is weak. It's been weakened. And what people used to do and did for at least a couple hundred years is they would take the scabs from someone who survived smallpox, grind them up into a powder, and snort that powder up the nose. And in that way, they were actually being inoculated with a weakened version of smallpox, which theoretically would not cause disease, but which would cause an immune response, which protected those people. But a very small percentage actually did get smallpox and die, right? So it's not a technique that we would use today, but back before we had vaccination, it was a reasonably, good technique to use, even though some people did die from it. So when it comes to microbes, we talked about how, the overwhelming majority of microorganisms on the earth are either beneficial or neutral to us, but they're very important for the ecosystem. Right? They're very important for the biosphere. And one of the most important things they do is to recycle nutrients. Because when a plant or animal dies, microorganisms decompose that plant or animal, and the nutrients are released back into the environment where they can be reassimilated by other living organisms. And this is especially true for nitrogen. The largest component of the air is nitrogen, and yet animals cannot get any nitrogen from the air. If only we could just inhale it and assimilate it, that would be great, but we can't do that. What happens is bacteria in the soil assimilate nitrogen, and then the plants can absorb that nitrogen, and then of course animals eat the plants, and animals eat the animals that eat the plants, and so on and so forth. So without microorganisms on the earth, there would not be this nutrient recycling. And so most life probably would not exist except microbial life. We can use microorganisms to clean up our messes. So oil has been leaking from the Earth's crust for millions of years. So there are microorganisms that have evolved to eat oil and use it as an energy source. What you see in this image are two technicians spraying rocks covered with oil with a mixture of microorganisms which will naturally degrade and break down the oil. There are also some microorganisms that can detoxify mercury, right, to make it much less toxic to humans. There's even a species of bacteria that can tolerate radiation that kills humans. So nature provides all these things, we just have to find them. There is an organism, Bacillus thuringiensis, which naturally produces a toxin which is toxic to insects. So the Bt toxin is something you can actually go buy at your local garden center or Lowe's or Home Depot, spread it on your lawn, and it will kill Japanese beetle grubs. They also genetically modify plants, some crops, to produce the Bt toxin so that when insects try to eat the crop, they die. It's a naturally occurring toxin in bacteria that is toxic to insects, and theoretically not to people. We've already talked about how we can genetically modify bacteria and use them as factories to manufacture things for us. We can also use bacteria as a lab tool to study genes and gene function, or what happens to the proteins when we mutate genes. So bacteria have done a tremendous amount for us in terms of biotechnology. Now, every living organism is colonized by microorganisms, and that includes humans. They live in us, they live on us. They cover your skin, they're on your mucous membranes, they're even found in a lot of tissues and organs. And they are natural, they're supposed to be there. The old term, which I still prefer, is normal flora, but flora means plant. And originally, bacteria were classified in the plant kingdom. So you still may hear the term normal flora because it flows off the tongue, but the new term is microbiota. And actually, a newer term is microbiome, which refers to all those microorganisms that live in us and on us and how they interact with each other and us. But you will still hear me use the term normal flora, but also normal microbiota. And because they're normal and beneficial, they do things for us like produce vitamins in our gut, like vitamin K and folic acid. If you're heavily colonized by good microorganisms, that helps prevent bad microorganisms from trying to colonize you and cause an infection. So normal flora are very important. Later on in the course, we'll talk about all the terrible things we do to destroy our normal flora, and that puts us at risk. So, as I mentioned, only a tiny fraction of all the microorganisms on the earth are actually dangerous to us, are actually pathogens. It seems like a lot, but relative to all the other microorganisms, it's just a tiny fraction of 1%. But one of the things we do worry about are newly emerging infectious diseases. So these can be something that's brand new, right, that we've never seen before, like HIV when we first discovered it, or Ebola. Or it could be a pathogen that already existed somewhere else, but is new to that particular country or continent, like West Nile is to the United States. When it comes to SARS, remember this lecture was written a while ago before COVID-19, but COVID-19 is a SARS. So there are two SARS viruses, SARS-1 and SARS-2. SARS-1, again, occurred in China back in the early 2000s, but it really didn't escape China, and they were able to get it under control, and it actually just sort of disappeared. SARS-2, which is also called COVID-19, is another version of SARS, and this one escaped China and spread all over the world and has killed huge numbers of people, and we'll talk about that later on too. So any questions? Because we're going to finish this lecture and then we're going to start the next. So no questions? Okay, the next lecture is on microscopy. So we've already mentioned that microorganisms are tiny. We need help to see them, and that help is with a microscope. So we're going to talk about microscopes and different kinds of microscopy, and then we're going to talk about staining. So just to review some measurements, who can tell me what symbol means here?
Micrometers.
Micrometer, right? A micrometer. So a micrometer is a millionth of a meter. That means you need 1,000,000 micrometers to make a meter. So it's very tiny. And we typically measure bacteria with micrometers. What's this unit here? Nanometer, right? So nanometers are 1/100000th of a meter. So that means you need 1,000,000,000 nanometers to make 1 meter. And you should all be familiar with the unit conversions from either 150 or chemistry if you've taken it yet. We're not really going to do a lot of that in this course, but just to refresh your memories. So the very first microscopes were created by a guy named Antony von Leeuwenhoek. And Leeuwenhoek was not a scientist. He just had an interest in lenses. And so he developed his own techniques for grinding lenses. He jealously guarded that technique, never revealed it to anyone, not even, right before he died. So he took that secret to the grave with him. It took people a long time after that to rediscover his techniques for grinding lenses, because he had come up with a way to grind superior lenses. His microscope was basically just a magnifying glass, but it was a superb magnifying glass. It could magnify up to 500 times. That's remarkable for a magnifying glass. So Leeuwenhoek was one of the first people, if not the first one that we know of, who actually saw microorganisms. The compound microscopes of the day, where they used multiple lenses, were not as good as Leeuwenhoek's microscopes. And Leeuwenhoek made a lot of them. He never sold or gave one of them to anyone. Again, he jealously guarded his microscopes. He would let people come look through them, but he would never give them away or sell them. Now today we use compound microscopes, so they use multiple lenses. Right? We have the ocular lenses, objective lens, condenser lenses. So there's multiple lenses involved, so they're compound microscopes. And of course you all know total magnification is equal to the objective times the ocular lens. How much does the ocular magnify? Right, 10 times, right? So 10 times. So we have 4 objective lenses. The lowest power lens, the scanning lens, is 4 times. The low power lens is 10 times. The high dry lens is 40 times. And the oil immersion lens is 100 times. So what's the maximum magnification we can get using this kind of microscope? What's the maximum? No, just the objective lens is 100, right? So Brigitte or Brigitte, I don't know how you say your name, but the ocular times the oil lens is 10 times 100 to make it 1,000. That's the maximum magnification, but that does require using immersion oil, which we're going to talk about. So just to review, if you have the scanning lens, which is 4x, and the total magnification would be 40. The low power lens is 10x for a total of 100. The high dry lens is 40x for a total of 400. And the oil lens is 100x for a total of 1,000 times magnification. So that's better than Leeuwenhoek's, but not a whole lot better. Now, in addition to magnification, remember that magnification is just enlarging the apparent image of an object and not actually making the object bigger, it just looks bigger. And so that's important. The other thing, resolution, is also very important. So resolution is the ability of the lenses to distinguish 2 points as separate. So when you magnify, when you enlarge the image, you must also be able to distinguish 2 points as separate. So the analogy that I always use, let's say you got 2 people standing side by side in the middle of the road and you're over a mile away. You can see there's some there, but it's not clear. As you slowly approach, those 2 people resolve into 2 individual people. So that's kind of like how resolution works, in that you need to be able to resolve 2 points as separate. If they're too close together, you just see one thing. Now, what determines the resolution and magnification is the quality of the energy source you use. So we use white light, for our microscopes. So of course you remember that light travels in waves, and so all EM radiation travels as waves. And so what is a wavelength? It's the distance from peak to peak, and that's the wavelength. So the longer that distance, the lower the energy. The shorter that distance, the higher the energy. So very short wavelengths are very high in energy. So the quality of the energy source you use is what determines how much you can magnify and how good you can resolve. Any questions? So now light does something funny when it travels through different substances or different media. So like when light travels through air and then glass, it bends. We call the bending of light refraction. So refraction. The refractive index is the degree to which a substance can bend light. So the problem with our microscopes, because they're compound lenses, your light goes from air to glass to air to glass to air to glass to air, and then glass again. So a lot of the light actually is bent away from the opening of the lens. Now, we call the openings apertures. And if you were to examine all the lenses carefully on a microscope, what you would find is that at increased magnification, the aperture gets smaller. So on the lowest power lens, the scanning lens, the 4X lens, there's a big, huge opening at the bottom of that lens, and that lets in a lot of light. But when you work your way up to the high, dry, and oil lenses, the opening is very tiny. So when you get to the oil lens, there's not enough light coming through the lens to really show the specimen brightly or clearly. So a way to compensate for that is to use immersion oil. Immersion oil has approximately the same refractive index as glass. So when the light goes through the slide, it goes through the oil and into the glass lens, that concentrates the available light. That makes the specimen much brighter and much sharper, so it's more clear. So that's why we have to use the immersion oil, right? Because light gets bent as it's passing from air to glass to air to glass, and too much light gets lost. So the oil helps to concentrate the available light. Now we're going to talk about the different kinds of microscopy, starting with the bright field, which is also called light microscopy. But the reason why it's a bright field is because the field of view is bright white, and the object appears darker against that bright field of view. So that's why it's called bright field or light microscopy. Now the organism we're going to look at for several of these types of microscopy is the same organism, so we can compare how the organism looks through different kinds of microscopes. Does anybody know what this organism is? Well, close. It is a protozoan, but it's not amoeba. This organism here actually swims around. Amoeba just crawl around. So another guess? Another type of protozoan? Nobody? Craig? Doreen? Melanie? Uriel? Aren't you lucky you're not sitting in front of me? So nobody knows, huh? Starts with a P. No? No guesses? How about a paramecium? Right, you've all heard of paramecium before. So this is a paramecium, so we use this as our example organism. So You can see some details here in the bright field. And when it comes to the internal workings of the microscope, don't worry about that. So next we'll look at dark field. So dark field illumination, the field of view is dark, and the specimen appears brighter against that dark background. So in particular, you can see some more internal structures here. So it's another way to view a specimen. And as we talk about each of these kinds of microscopes, we're going to be increasing in complexity as well as cost. So these microscopes get more complicated and more costly. With phase contrast microscopy, this is actually really cool, and we do have phase contrast microscopes in our labs, and normally I would demonstrate and show you, how these work. Because you know that light, white light is made up of many different colors, right? And what do you use to see the different colors of light? I know you've all seen that. Shine light through thing and it splits it into all the colors of the rainbow. A prism. A prism. And yes, Sarah, spectrophotometers do have prisms in them. But the prism can separate light. When you separate light into different wavelengths, that is called diffraction. So refraction is the bending of light. Diffraction is the splitting of light into different wavelengths. So a phase contrast scope splits the light into different wavelengths. So specimens, or different parts of the specimen, absorb different wavelengths of light. So you get a contrast. And in particular, in this image, we can now see something in here much more clearly than we could see in the other two images. So what can we see on this paramecium much more clearly than we can in either of these two images? So what shows up now? Phase contrast.
The outside.
Yeah, but what's on the outside?
The cell wall.
No, not a cell wall, but the cilia, right? So Ivy said the cilia, right? So we see the cilia here. Now we can clearly see them in the phase contrast scope. So the phase contrast is really cool, especially when you're looking at live organisms. And I would normally show you these in lab, but if you go to my web page, I have some videos of amoeba that I took with the phase contrast, and I actually have a video of an amoeba exploding. So it's pretty fun to watch amoeba death. Moving on to fluorescence microscopy. This is similar to dark field because the field of view is dark, but in this case we're going to use fluorescent dyes. And the fancy name is fluorochromes, but fluorescent dyes. And when you shine UV light on them, they glow, right? They fluoresce. And you're all familiar with fluorescence. You've all bought, fluorescent markers or binders and notebooks with fluorescent covers on them. They're very bright because they're actually re-emitting some of the light that they're absorbing, and they fluoresce, so they kind of glow. Now, usually how this works is you can use this as a diagnostic tool, say to identify a pathogen, and you need to use antibodies. You probably heard about antibodies in the news because they're talking about them with regard to immunity to COVID. But antibodies are special immune proteins that especially stick to other proteins and polysaccharides. So you have antibodies to a particular pathogen. You attach the fluorescent dye, right, to the antibody. You take a specimen from the patient, smear it on a slide, apply your fluorescent antibody to the slide. If the antibody sticks right, to the specimen, then that's a positive identification, a positive test result. In this image here, we're seeing that these spiral-shaped organisms are all lit up and they're glowing green, right? Because of the fluorescent dye that's used, they glow green. So it's easy to spot these glowing cells against a dark background. In this example, they're probably using something like Treponema pallidum, which causes syphilis. But if you're doing research, cell biology research, maybe you want to see if your cells are producing a particular protein, or maybe where that protein is located inside the cell. Again, you can use a fluorescent antibody, which will bind only to that special protein. You add it to your cell culture, and then zap it with UV light and see if there's any glowing anywhere in the cell. So you can use fluorescence microscopy as a diagnostic tool or to look for proteins being produced inside of cells. Confocal microscopy is similar to fluorescence microscopy. We're still using fluorochromes, but instead of UV light, we're using laser light, right, to excite those molecules. The laser allows us to generate a three-dimensional image because you can scan the specimen with the laser. Once again, we're looking at a paramecium, and we see two very interesting star-shaped structures on the inside. So now we're going to review your Biology 150 knowledge, and we're going to review tonicity. Remember tonicity? Remember hypertonic, hypotonic, isotonic? So paramecia live in fresh water. What's the tonicity of fresh water? So what's the tonicity of fresh water? Hyper, hypo, or isotonic? I got two hypos. Everyone agree? You should, right? Because that's correct, right? So fresh water is hypotonic, so that means it has a low concentration of solutes. So where does the water want to go? When you put a cell in a hypotonic environment, where does the water want to go? No, but where does the water want to go? Right, inside the cell, right? Because the cell is going to have higher concentrations of solutes on the inside. So water moves into the cell. What happens to that poor cell? Yeah, it explodes, right? So how did the paramecia survive in a freshwater environment? So how do the paramecia survive in a freshwater environment? Why do they not explode? Thicker wall? No, not really. Remember, it's not a plant cell, so there's no cellulose to reinforce the cell wall. So how else might this paramecium survive? So I know we do talk about this in 150. Nobody remembers? So these structures are contractile vacuoles. As the water moves into the cell, the vacuoles absorb that water, they swell up, and then they contract and squirt the water back out. And what we're seeing here is the contracted state. But all paramecia have contractile vacuoles that absorb the water that's constantly moving into the cell, right, by osmosis, and then it squeezes the water back out. So that's how paramecia can survive in a hypotonic environment. So don't forget all that stuff you learned in 150. Moving on to electron microscopy, electrons are very high energy particles. They have very short wavelengths. Also, what's the charge of an electron? Negative, right? So electron beams are shot through a specimen. So in the case of transmission electron microscopy, you're actually going to look at a cross-section of a specimen. So what you need is called an ultra-thin slice, super thin. The problem with the super thin slice is that all the electrons are going to go right through it, and you won't see anything. So they have to use a special stain, a heavy metal, OS. What element is OS on the periodic table? And Gareen should have the periodic table memorized, right? Yes, sir, it's osmium. It's osmium.
That one. I wasn't sure about that one.
Yeah. So osmium is a heavy metal that's used to stain the specimen. And because there are parts of the specimen that are naturally more dense and less dense, there'll be more osmium and less osmium. So some of the electrons get through, some get stuck, some bounce off, but you can develop an image of contrasts. And in the old days, they used a photographic plate to develop the image. These days, it's a computer sensor that actually develops the image. But the images are technically in black and white. When you see a colorized image of an electron micrograph, it's fake. It's been colorized. Those are not the natural colors. So here we see a cross-section of the paramecium. So this is actually a slice through a paramecium. So we're talking about, an ultra-thin slice. It is not green and brown as shown. That's just been colorized to look that way. But look at the magnification, up to 100,000 times versus 1,000 that we can see. 100,000 times. And the resolution, 2.5 nanometers. So that means if two objects are at least 2.5 nanometers apart, you can see two objects and not just one. You do see a bunch of objects inside. What's the general term for all those objects inside? Organelles, right? They're organelles. So in our microscopes, we can see little tiny dots floating around, but here you can see much larger objects. You can see the organelles. And no McCormick College does not have any electron microscopes. Now we can also take the beam of electrons and use it to scan the outside of an object. And so we can develop a three-dimensional image of an object using scanning electron microscopy. So here's the paramecium again, but it's the outside of the paramecium. Now what can you see clearly all over that paramecium?
The cilia.
Right, the cilia. And no, it's not that greenish yellow color, it's just been colorized. Now, when you scan a specimen with a beam of electrons, you don't get as good of magnification or resolution, so around 10,000 times and about 20 nanometers. So, but you get beautiful exterior three-dimensional images using a scanning electron microscope. So we're getting more sophisticated, more expensive as we go along. We have scanning probe microscopy, things like scanning tunneling microscopy and atomic force microscopy that we'll see in a second. But what I want to impress upon you is that we now have the ability to see down to the atomic level. We can actually see how atoms are arranged in molecules now. Only with the help of computers, but we can see these things. So what we're looking at here, all these little bumps and balls are actually atoms. We see how they're lined up. So incredible resolution. So it's truly amazing what can be seen today. with extremely powerful microscopes and extremely powerful computers. We can use atomic force microscopy, which measures the forces between atoms and uses those data to create a three-dimensional image. But again, I just want to impress upon you that we can now see how atoms are arranged in molecules. So no more playing with balls and sticks. I don't know what they do in chemistry anymore, but they don't really need to use balls and sticks anymore, right? So now let's talk about something that would have been fun to do in lab, but we're not doing that. But you have to make slides of bacteria and then stain them. So the idea is you're going to spread a very thin film of bacteria on the surface of a glass slide, and that's your smear. You let it dry, and then you heat fix it. And you heat fix it by passing the slide through a flame. And that actually helps the bacteria to stick to the slide. It does help kill them, but the stains are going to do that anyway. The main point to heat fixing is it helps the bacteria stick to the slides. If you don't heat fix, you're adding liquids, you're rinsing with water, you're blotting the slides. If you don't heat fix, you're going to lose some of your bacteria. But even worse, when you go to look at the smear under oil, the bacteria actually lift off the glass and start floating through the oil. And that makes it hard to see them. So after you make your smear and dry it, you always heat fix it in the flame. Then you can stain it. Now, the reason why we stain, especially bacteria, is that bacteria are tiny, and even if they produce a pigment, it's usually not enough to help us see them. So bacteria are essentially colorless and they have no contrast. So it's very, very hard to see them if they are not stained. And if we were in lab, what would happen is someone would have a very weak stain, and when they go to put the oil, all the bacteria disappear, right? Because they really need that stain to give them color, to make them darker so we can see them. But as I said, we have to kill the bacteria to do that. Trying to look at live organisms that are stained is extremely difficult because almost every stain is toxic to live organisms. So it's really hard to look at a living organism that is stained. Now, if we were in lab, when we did motility, one of the things that you had to do was to actually find and focus live bacteria that are not stained. And that can be very challenging because the bacteria have no contrast. You're trying to find them without any color. But it is something that was fun to do, and most students did learn how to do it. But we need to put stain on the bacteria so we can see them. Now, I'm not going to go into a lot of details about how stains work. The main idea is that the stains are charged. They're either positively charged or negatively charged, and the cells or parts of the cells are charged. Opposite charges attract, and that's why the stains stick. So if the stain sticks, in general we say that's a positive stain because the stain sticks. This gets a little confusing when we start talking about the gram stain and gram positive and gram negative. But the main idea is that stains stick to cells or to parts of cells because of the charges. Now, what do we call a negative ion?
Anion.
An anion. And what do we call a positive ion?
A cation.
A cation. And I always make the joke, where did you go this summer? On a vacattion or a vacation, right? But you always stain bacteria to give them color. Now in lab, as your introduction to staining and slide making, we would start off with a simple stain. A simple stain, you just use one dye, something like crystal violet or safranin or methylene blue, just to give the bacteria some color so you can see them. And all a simple stain tells you is the shape of the bacteria and the arrangement. Sometimes bacteria are stuck together in characteristic arrangements like clusters or chains, and so you can see that when they're stained. That's not useful information. That's not good enough. So, in fact, after you do a simple stain, you'll never do it again. What you will do is a gram stain, because that's the all-important stain, is a gram stain. So a gram stain is a differential stain. We can differentiate gram-positive bacteria from gram-negative bacteria. And what makes bacteria either gram-positive or negative is the type of cell wall they have. And that's why we do a gram stain. We want to know what kind of cell wall those bacteria have. Knowing what type of cell wall tells us three things. It can tell us the best antibiotic to use. Because, for example, penicillin only targets gram-positive bacteria. So if you had a gram-negative infection, you would not use penicillin. Knowing what kind of cell wall the bacteria have can also indicate the possible source of contamination. Where did the bacteria come from? Let's say you go out to a restaurant, and then a few hours later, nausea, vomiting, diarrhea because you got food poisoning, and it's a gram-negative rod. Well, that means that restaurant employee did not wash his or her hands after going to the bathroom. And lastly, knowing what kind of cell wall the bacteria have is the first step to identify an unknown bacterium. You want to know, is it a gram-positive or negative rod, coccus, or spiral? That's where you start. So you always do a gram stain. Not only will that tell you shape and arrangement, it tells you what kind of cell wall the bacteria have. And because you know that, you can pick the best antibiotic. You can help determine where that organism came from, its source. It's also the first step to identify it. But that's why we always do a gram stain. We just let you play with a simple stain to get practice making smears and more practice with the microscope. But after that lab, you never do a simple stain again. So here's the gram stain procedure, and we'll talk more about this next week too in lab. So there are multiple steps to the gram stain, and you always have to do all 4 steps right to every smear that you make. The first stain, the primary stain, is crystal violet. If you were to examine the slide right after adding crystal violet, you'll see that everything is purple. But then you add iodine. Iodine reacts with crystal violet to form a complex that sticks to the gram-positive cell wall, but not the gram-negative cell wall. Then you decolorize using 95% ethyl alcohol, also known as ethanol. And the 95% ethyl alcohol removes the excess crystal violet from gram-positives and all the crystal violet from gram-negatives. So at this point, the gram-positives are purple, but now the gram-negatives have no color. And as we said, bacteria without color are very hard to see. So we counterstain with safranin, a red stain. The gram-positive bacteria are not affected by the safranin because they're already purple. But the gram-negative bacteria, they pick up the safranin, so they appear red or pink. So gram-positive bacteria are purple, and gram-negative bacteria are pink or red. And that's how you tell which one's gram-positive and which one's gram-negative. Of course, there are problems with stain, and we'll talk more about those problems next week in lab. But here we see an image of some gram-stained bacteria, and we see an example of one of the important problems. So if you see right here, we have a chain of 4 cells. 3 of the cells are purple and 1 is pink. If it's a chain of bacteria, how can that be? If we look at this cell here, this cell is both purple and pink. The same cell. So how can that be, right? That doesn't seem right. And so here's one of the problems with gram-positive bacteria. As they get old, as they age, their cell wall begins to break down. It begins to deteriorate. That means the gram-positive cell wall can no longer hold on to the crystal violet that well. So when you decolorize with the 95% ethyl alcohol, you actually wash away almost all the crystal violet, and so the cells look pink. You can believe that's really confusing if you're making your stains, and especially if you're doing it unknown and you're using old bacteria, you don't know whether they're supposed to be gram-positive or gram-negative, right? But that is one of the problems that does happen, where as the cells begin to age, they become what's called gram-variable. And even though they should be purple, they may look pink or be half pink, half purple. So that's one of the problems with the gram stain. We'll talk about more problems next week. So the acid-fast stain is another stain that we do in lab. And the acid-fast stain uses carbol fuchsin as a primary stain, and it's fuchsia colored, a kind of purplish pink or reddish purple, color. And the counter stain is methylene blue. So you use this specifically to look for a certain pathogen. So this image here is a sputum smear. So sputum is mucus from the lungs, and it's been stained with the acid-fast stain. What you hope to see is all blue. All blue is a good thing. If you see these red fuchsia-colored rods, that's bad, because the pathogen that is acid-fast positive is Mycobacterium tuberculosis. So this patient has active TB, right? So you don't ever want to see any, fuchsia-colored rods in your sputum, because that means you have TB. Everything ideally should be blue, right? That's not acid-fast. So acid-fast means that when you use the acid alcohol to decolorize, if the organism is acid-fast positive, it resists decolorizing with the acid alcohol. So it holds on to the stain. It's acid-fast. It resists decolorizing. In lab, we use a more harmless version of this organism so you can actually see what an acid-fast positive organism looks like. So some special stains, or at least your textbook calls them special stains. Here we have the negative stain. So remember I said that in general, if the stain sticks to whatever you're trying to see, that's a positive stain. But there are certain techniques called negative stains. And so the negative stain means that the object or structure you're trying to see is not stained. Everything else is, including the glass slide. The actual structure you want to see is not stained. As a result, you can see it. In this example here, we have a capsule stain. Some bacteria produce a thick polysaccharide capsule. So the special stain used here does not stain capsules. It does stain bacteria, it stains the glass, but not capsules. So you can see all these clear zones around the stained bacteria. Those clear zones are the capsule. We can see them because everything else is stained, but not the capsule. So we call that a negative stain. And again, the terms positive and negative can get a little confusing when you start talking about gram-positive and gram-negative. But in general, if the stain sticks, it's a positive stain. If the stain does not stick to whatever you're trying to see, that's a negative stain. Another special stain is the spore stain. So certain bacteria produce endospores, which eventually get released when the cell dies, and they're just called spores. So the traditional stain used a malachite green, and you have to use heat to make the malachite green penetrate the spore coat because it's very thick and tough. And it's hard to see in this image, but there are little green dots inside of cells, as well as some that have been released and some free spores. So this is a traditional staining technique where the spores appear green or maybe kind of bluish green, and the cells are stained red with safranin. And then flagella, if bacteria can swim, they use flagella, only flagella, nothing else. And the problem with flagella, since the bacteria are microscopic, they're already tiny, the flagella on them are even smaller. So there's no way to see them with an ordinary microscope unless you use a special stain. And I use the analogy of mascara. So ladies, what does mascara do you? Or maybe some of the gentlemen, but what does mascara do for you?
Makes your eyelashes longer.
Yeah, it makes them longer and thicker, right? Does it really make them longer and thicker? No, it's just a bunch of goop you put on your eyelashes, which is mostly charcoal and fats, but to give the appearance that they're longer and thicker, right? So the special stain, it does the same thing. It makes the flagella appear thicker so you can see them in the microscope. So any questions on staining? I know you weren't expecting a lecture on makeup, but no questions on staining? Okay, so we're going to stop here for lecture. We'll start lab at 1:20. So you can take a 10-minute break, and we'll meet back for lab at 1:20. Okay?
Thank you.
Right, so I'm going to close this session so I can open up the lab session.